our topic today. We'll talk about the COVID infection, of course, and we'll try to analyze data, gene expression and proteomics expression data in order to try to find targets, drug targets, and possible treatments. And we'll use the tool called Genome Enhancer, and I will introduce you a little bit in that. So basically, what we are going to do today is first we'll take transcriptomics data, and there is some data available already. So there's this paper which was published a couple of days ago, pre-published, I would say. And the data was actually recorded in this gene expression omnibus on infection of lung epithelial cells by coronavirus of this COVID-2. And then we will take this data, and we will take also the proteomics data from another recent paper where the cells were also infected by coronavirus, and the mass spec proteomics was done. So basically, it's a lot of data, and then this data will be, so what we did, we actually put this data into our tool called Genome Enhancer, and I will explain how it works. And then this Genome Enhancer does actually analysis of this data, and at the end it predicts most important or most promising drug targets. Okay, so some people reply that they cannot hear. So technical support will basically help you. Okay, but most of the people, I hope, can hear me well. So the structure of the seminar today will be, so I will make a little presentation about the approach. Then I will go online to show how this data looks in this software, and please, if you have any questions, just write here in chat, and then at the end of the seminar, which will be like 40, maybe 30-40 minutes, I will try to answer those questions which you wrote in the chat. So I will be online for another maybe half an hour to answer those questions. So don't be in a hurry. Just prepare your questions and put them on, in this chat. So if some questions will be not answered, so we'll be able to answer them later, either by emails or in another seminar like this. Okay, so just coming back to my slides, basically this is the pipeline we are going to do. So first we will try to find the targets, the drug targets, and then from those drug targets we'll propose the drugs or medicaments which basically have a potential to be used as a treatment. And of course here we have to put in a disclaimer or warning that all these results are done just with computer modeling, and of course further validation of the identified drugs has to be done prior to use it in any clinical practice or treatment of the patients. So it's pure research for so far. Okay, so the Genome Enhancer works like so from the name, because it's enhancer, Genome Enhancer. You can guess, and basically we pay attention to enhancers in genome. And we analyze the so-called dark matter of genome. So those regions of genome which are located between the genes and which are responsible for gene activation. And in the infection or in other diseases, when we see actually many genes are changing their expression rapidly, and we will see this in the case of this data. Something happening, so basically something happening in the regulation of those genes, and they are changing their expression. That's why the primary goal of this algorithm is to look between the genes into the enhancers and try to model from those enhancers. Okay, and then So what we do, we actually check in the enhancers in the regions near the genes which are differentially expressed. We check those genomic regions and try to build a model from transcription factor binding sites. And here we use machine learning. I will introduce you a bit later how we do that. But at the end, actually, of that learning, the system comes up with a composition of transcription factors which are responsible of activating those genes. And they are presenting in these slides as these little ROMs. You know, we have this composition of factors. And then the next step, which algorithm is doing, is actually asking question whether there is a certain master regulator in the signal transduction which activates those transcription factors altogether. And that's done using the signal transduction network and the algorithms of searching for the shortest passes in this network. And then at the last step, once we find those master regulators, we call them master regulators, we try to predict which drugs can be used to inhibit or to actually manipulate those activity of these master regulators. And from those, this cascade actually assuming that after inhibiting those master regulators, we basically can change the activity of those genes which are changing their expression in the disease or upon infection. So I hope, yeah, there's, I see there are some question concerning the audio, but I hope you'll find a way. Yeah, technical support will help you, right? So, okay. Okay, going on further. So having such algorithm in mind, then the system is able to integrate various types of omics data. In here, in this case, as I introduced in the beginning, we will use transcriptomics and proteomics data. But generally, the algorithm is capable of integrating several types of omics data. For instance, this transcriptomics, RNA-seq data, even single cell, and proteomics data, and also epigenomics data, for instance, ChIP-seq or DNA methylation data or toxic data. And the way how it is done, the integration is done, is actually basically on the logic. It's based on the logic of searching for master regulators. So as you can see from this picture, the list of differentially expressed genes are coming from transcriptomics, but then proteomics, if it's done in a similar system or actually optimally in the same samples, actually detecting which proteins are present or changing their activity or changing the amount in the system, and then the algorithm which searches for the master regulators makes the way through the network, giving priority to those proteins which are observed in the proteomics data. So that's how these transcriptomics and proteomics are integrated. There is also a possibility to integrate epigenomics data when, for instance, if you have ATAC-seq or ChIP-seq data, which give us information about open chromatin in the genome, then the algorithm will take these regions into account, giving more priority to those regions when searching for transcription factor binding sites. If these data are available, they will be included by the algorithm and will improve the quality of the predictions. If they are not available, it's also fine, like in this case. So we are still waiting for epigenomic data to come for the COVID infection. So the algorithm by default will take the just 5' regions of the differentially expressed genes as a kind of proxy of the regions which are place a lot of regulation for those genes. Okay, And at the end, actually, you will get the system will generate several results comparing transcriptomics and proteomics data, also making those nice diagrams about signal transduction cascades, and also plotting those data on this signal transduction cascade. So you will see this online. And at the end, as outcome of the system, we come up with a mechanism of regulation of those genes, and based on that those mechanisms, we propose the best drug targets, also biomarkers, as well as potential prospective drugs. So just a few words how basically the transcription factor binding sites are predicted, and that's a core of the algorithm. So as I said, we are analyzing the places, regions between the genes. So we have to find how those regions are regulating the genes, and they are regulated by transcription factors. So we have to predict the transcription factor binding sites. And how we do that, we first collect known binding sites for a given transcription factor, for instance, AP1, and that's done for many years in our database called TransFac. And then we build a model, a consensus of those transcription factor binding sites, also a more generalized way of presenting the consensus in the form of this positional weight matrix, which gives some weights to different nucleotides in different positions. And then this matrix, this consensus, is applied to the regions of our interest in genome to find new potential binding sites for these transcription factors. And having a lot of transcription factors in our library, and basically these are several thousands, we can predict binding sites for many different transcription factors. Okay, and but enhancers are more complex structures. So basically, if you look at this, for instance, enhancer of an interferon beta gene, then you can see that this enhancer is composed of several transcription factor binding sites, which are sitting together in a close proximity. They help to bind various different transcription factors and regulate this enhancer by joint efforts, I would say, right? So by synergy, these factors are binding to DNA and also bind to each other, and they make a complex which some people call enhantiosome, and this complex is actually regulate the activity of the genes. And in order to find those composite modules, we call them, or composition of transcription factor binding sites, the algorithm here we call it machine learning algorithm from the area of genetic algorithms is applied, which finds these combinations automatically. So it takes all the regions of interest from the genes which are differentially expressed, and then it actually tries to find the best combination of transcription factor binding sites. So this is the core of our algorithm search for the master regulators. And then actually the search for the master regulators, as I have mentioned to you, is done through this signal transduction network search, when the transcription factors, those which we found by the composite module analyst, are used, and then in the signal transduction network, the search is done until we find those master regulators. Moreover, in different diseases, like for instance, in the response of the cells to infection, or in inflammation, as we will talk about this later, this regulation is really changing. So instead of initial master regulators, the system finds new master regulators with so-called positive feedback loops. So we'll see them later in our analysis. So which means that actually this regulation support each other, support itself. So these master regulators, they activate those transcription factors, which are in turn regulate the genes which encode the master regulator itself. So it starts activating itself through the positive feedback loops. And when system has a lot of those positive feedback loops, it goes into the circle and basically goes into the disease mode. So that's why our algorithm, while searching for these master regulators, also checks whether these feedback loops are present there. And once we find this, then those top master regulators which have, which are involved in these feedback loops, they can be considered as the best drug targets, and these respective genes are the best prognostic biomarkers. So as I said, this positive feedback loops is similar like we observe in severe inflammation. It's like in fire. So when the fire itself supports itself, and it goes more and more like a fire. That's search for these positive feedback loops is essential for finding the best drug targets. Okay, so with that, I will go basically online to show you how it works. So I will share the screen, just a moment. So share. Okay. So as we discussed, actually, there were these two sets of data, which were transcriptomics and proteomics data. Let me first show to you these data files. So you see them now on the screen. So this first was proteomics data published in this paper, which I showed to you. And that's a very nice mass spectrometry data, which where they measured amount of proteins in infected cells after 2 hours, 6 hours, and so forth until 24 hours, and there's a control cells. So this is the first set of data we're going to use. And the second set of data are coming from RNA-seq, from this paper published just recently of a group from New York, where it's typical RNA-seq results where you see these counts, so basically how many reads were identified for a particular gene in different settings. And in this paper, they did infection not only with coronavirus, but also with other viruses, and they are comparing them to each other. We here are just taking the data from coronavirus. So there were 3 replicas of coronavirus infection, and actually the control cells, and there was also the time point was 24 hours after infection. So we have these two data sets, and now we are going to actually load them into the Genome Enhancer. So the Genome Enhancer looks like this. So we'll discuss it maybe later, how this subscription and how it works. But once you're in this Genome Enhancer, so you can create a project. Like I created this project called COVID-19 drug targets, and you can put here your data. So just by drag and drop, basically you have transcriptomics data, you drag and drop here. You have proteomics data, you drag and drop it here, and then you have to import it into the system by uploading it. So it detects the format. It checks that it's a transcriptomics data because the first ID is a gene symbol, and you can import it. And then you actually import proteomics data. So it detects the first ID, which is a UniProt ID. So it identifies it's a proteomics data. So you can also load it. So we will discuss maybe later about acceptable formats. It's not only tables, but could be any kind of raw data as well. But now since we have already pre-processed data, it's easy to start with the tables. So next, We have to take this data and look what kind of information we have in this data tabs. So this is the transcriptomics data with all the columns in it. And here the proteomics data with all the columns in it. So we take this data into our workspace and then we go next. And now it creates to me two boxes where I would call the first box, for example, COVID-19, and second box just control. So, and what I'm going to do, I first take the COVID infected data, put it in this box, the control, putting it in this box. This is transcriptomics, and the same for proteomics. For proteomics, I will take 24 hours only for the infected cells, so to have the comparable data, and then the control also after 24 hours. So just putting him in the box. So now we have these two categories, COVID infection and control. We go next. And now we would like to compare them. So basically we take the COVID infection category and compare them to the control. And now we have to select basically the disease we are going to analyze. And the reason is that we will first, the system will predict the enhancers, it will do this master regulator search. But then after finding the master regulators, it will search for the drugs. And to make a best fit to the drugs which are available and which are actually tested for a particular disease, we have to select the disease we are interested in. And first we, of course, here are interested in virus diseases. So it's, we have this coronavirus infection as a disease, but we would like to extend this category a bit broader to all RNA virus infections. So just virus diseases, to also look to other drugs, not only those which are already available for coronavirus, which is none, I would say, but also a bit broader to have a possibility to do kind of to search for broader categories of the drugs. Also, we are interested to treat the disease itself, so the symptoms. And as you know, the most important symptom, and it's also in the name of the disease, severe acute respiratory syndrome. Also, we already know that, in fact, some of the drugs are already repurposed from other types of virus infections. And so we can take it here as well. So to look at those drugs as well. For instance, HIV infection or hepatitis infection. That we can also take here as a potential drug candidates, which we look at. Okay, that's it. So basically we prepared it and now we just start analysis. So this analysis involves quite a number of kind of complex steps, including the search for enhancers, machine learning. So overall, it takes like 4 or 5 hours, depending on type of the data we use. So of course, we are not going to sit and wait for these 4 hours now. So that's why I would basically go to back to the project and select the project which I already done. And just a little thing here, I'm reading also comments. So all the questions I'm going to answer at the end of the presentations. Well, maybe not all, but I will check what kind of questions we can answer. So there will be enough time. So, okay. Okay, so this project, which I have already run before, with the same set of data, so transcriptomics and proteomics, I put it twice here. And so we can look at these reports which it generates. So that's after 4 or 5 hours of the work, it generates these two reports. One is called Brief Tale, which is a little summary of everything, and another is full story. So let's look at the full story report. So The report is prepared in the form of scientific publication. Basically, it's just to help you to find everything you need for your further publication, because often you need some parts of the methods and everything. So the system puts everything in such a form. So it actually reports to you that out of all the work which the algorithm done, first it detects these three transcription factors as most important transcription factors involved in activation of the gene upon infection. And from this transcription factor, it also then finds that these two signal transduction molecules, like interleukin-1 receptor and the kinase PLK3, they are basically responsible for the activation of these transcription factors and might be the most promising drug target. Okay, let's go through this report. As you can see, it summarizes the method, how it is done, also presents the data which we generated. Sorry. So the COVID and control. And first, of course, it takes the transcriptomics data and start analyzing, comparing the infected cells versus control. And then it detects actually pretty high number of differentially expressed genes. So more than 1,000 upregulated and 6,500 downregulated genes. But this is with pretty soft cutoffs, like with p-value cutoffs. But of course, it calculates all false discovery rates and also identifies the most significant upregulated genes and most significant downregulated genes. So you see here the top genes which are upregulated and downregulated. And then it takes the whole set of genes and tries to build basically the heat maps to compare the infection and control. And you see clearly they are separated. So the infection is clearly make the cells completely different from control. So they are separated. Then the upregulated genes are mapped to the gene ontology. That's a standard step. And we want to know actually what kind of functions are changing in these cells. And the biggest change you see clearly here, the innate immune response is changing in those cells, the genes belonging to those response. And cytokines, we'll talk about them in that respect. And here we take those genes and map them to various standard pathways. And then you see those pathways related to immune response, like TNF alpha pathways and interferon gamma pathways. They are activated, and NF-kappa B pathway, they are activated. So this we clearly see the immune response genes are activated. And in fact, even here we can say that not so much of the infection itself, but rather a lot of immune cell response, which we see here. Then we take those genes which are differentially upregulated and basically try to see how many of those genes are already known biomarkers for different diseases. And of course, it was a bit surprise, but now it's clear why, that what we see here, that a lot of those genes which are upregulated belong to the immune system diseases, to the autoimmune processes, even to the neoplasm, to inflammatory, and the diseases related to skin. So that's very important to take into account. So concerning the downregulated genes, you see the same pictures. And then now it takes the proteins and try to identify differentially expressed proteins. And actually, a number of proteins were identified. And interestingly, that those proteins are in fact quite different from the from the transcripts which we observe. So we see different categories which are upregulated in the protein level. So maybe what we see here that it's actually time difference. So the transcripts are already changing, but on the protein level, it's too early to see those changes. And we see the bigger changes related to the translation machinery and to those proteins which are involved in metabolism of the cells. So when we compare, when the system compares purely transcriptomics with proteomics data, it shows no correlation. And that's not a surprise in such a quick processes like infection. It's very often we see no correlation between proteomics and transcriptomics data. Still, it means that both data sets brings their own information into the analysis, and they are not they are actually supplementing each other. So you can also see that when we compare transcriptomics and proteomics data, then there is very little overlap between those sets. But now we want to find the enhancers, right? So we want to find those composition of transcription factors which are upregulating our genes. And this algorithm which I have introduced is now was working quite hard and identified the composition of these few transcription factor binding sites, which happen to be found in the promoters of upregulated genes in different types of clusters. And you see these factors like NF-kappa B and IRF1 and Jun and Jun SMAC. So clearly transcription factors which are involved in, again, in immune cell, in immune response and inflammation. So there is also certain statistics showing how those clusters are different from the background. So the genes which have those clusters of factors are presented here. Now we go further to find for the master regulators in these networks, starting from transcription factors, and this is a list of those master regulators. But let me just show it on the diagram. So I will make a zoom of that. So this is a diagram of the signal transduction cascade, which is predicted to regulate upregulated genes. So if you see here at the bottom, you see these boxes. They represent the upregulated genes, which are in turn are regulated by these transcription factors, which are in blue, and these in these boxes showing the transcription factor compositions. And then this transcription factor activity is regulated through this big cascade of signal transduction events. And at the top of that cascade, we see these complexes of master regulators, and you see them like interferon alpha and beta, complex together with interferon receptor. Then we see interleukin-1 with their receptors. We see actually TNF alpha. So those cytokines and which are really involved in inflammation. So there are other components which belong to NF-kappa pathway. So now here you can also see that these little shades around the nodes represent the transcriptomics data. So at the left, you see the transcriptomics data. At the right, you can see the proteomics data. Sometimes you see that on the transcriptomic level, this protein is upregulated, but on the proteomics level, it's downregulated. That's so, but important, it's actually involved in this regulation. So now we know this mechanism, so we got the mechanism, and we go to the next step to identify potential drugs which may be targeting those drug targets. And so first we look at the system looks through all the master regulators, which were identified, and detects those of them where a certain number of drugs are already known. So for example, there is 3 drugs from different types of diseases which are already known for inhibiting interleukin-1 receptor 1. And as for TNF, there's up to 30 different drugs which are known. But not all of them actually are really tested for a given disease, so for the infection. So what is present then in the next table is actually the final table of those drugs which are ranked at the end, is a ranked set of drugs which shows you, first of all, that this particular drug is known to be actually affecting those targets which are from our list. And this drug is already approved by the FDA, or it is going through a certain step of clinical trials over the disease we are studying, or diseases we are studying. So you remember we selected different diseases related to viruses, to HIV, to hepatitis diseases, and for example, interferon, and you see these 3 types or even 4 types of drugs which are related to interferon are used in the clinics, actually used in different phases of clinical trials, and some of them are already used in real clinics to treat the disease. But what is interesting is actually what we can detect further. So, and one of the top candidate which we here see is this drug called Anakinra, which is actually not yet tested on all clinical trials phases for any of the virus diseases. So there is some phase 1 only for it. But the main target of that drug is actually, it's used in rheumatoid arthritis and other type of diseases related to inflammation. And the target of it is actually interleukin-1 receptor 1. So the system finds this as an interesting candidate for further consideration. Actually, this is the top 5 candidates. We can have a look at the bigger table. So we see here that this Anakinra, and there is a next drug which we had also discussed in the beginning, poietin alpha, which can also not yet actually considered by FDA and not gone through all clinical trials, but could be actually interesting candidate. There is other sets of drugs which are present here in the list, and we can look at them as well. So actually going further, there's a bigger list of drugs which actually considers the whole set of targets, but have nothing to do maybe with this disease, so they may be also interesting candidates. But what is next interesting for us in terms of basically searching for new drugs or for drug development, so as a research project. So there is a possibility to look at those sets of different compounds, active compounds, which are available as compounds involved in different drugs already on the market, or just which are part of different clinical trials. And in the system, we used a library of 13,000 different active compounds, and we applied a chemoinformatics tool called PAS, which predicts for those compounds potential drug targets. And then our system, Genome Enhancer system, takes those predicted targets for those compounds and interlink them with, actually cross them with our predicted drug targets with the master regulators. And after doing that, and also checking whether this drug could be also potentially antiviral or have antiviral potential, we come up with a list of potential candidates which might be used for drug repurposing. Okay, so at the end, this report, we see a little summary and also a proposed mechanism and also a proposed combination of drugs. It's actually a pure research idea of, if this mechanism of response of the cells to the infection involves not just one master regulator, but several of them. So possibility to inhibit several of them as a set would be an interesting idea. So then this algorithm searches for the best combination of drugs and actually active compounds, which may be altogether acting against several drug targets, not just one. So this report also contains description of the methods and all the needed information which can be used for the publications. So you see this report is pretty full of a lot of data. So also actually more clear search can be done by looking at this table more in detail, looking at the bigger sets of the data. But just in the conclusion of this presentation, I will go back to my slides. Actually, let me show this one. Just to mention that the transcriptomics data, when we started, yes, I think it's shared. Yes, can you? Yeah, I guess you see. The transcriptomics data, when we started to analyze it, were still not published. It was available gene expression omnibus, but just a couple of days ago, this publication came up just from the authors of the data, and they actually observed this that indeed a lot of inflammation and a lot of immune response genes are activated in this system. And as for the predicted drugs, which I showed in the results, so concerning this Anakinra, of course, it's an interesting candidate, and there is already consideration, and in some clinic, perhaps it's going to be considered. The problem is that in severe cases, the infected people, they got sometimes and pretty often in severe cases got a so-called cytokine storm, so which is similar to what is observed in sepsis. And in such a situation, the immune system has to be blocked. And this Anakinra is known antibody which is used to block immune system in such cases, and it blocks it through interleukin-1. There are other candidates which were also in our list, which are, as we know, already going now into clinical trials, and maybe we can touch it in next time when we talk about that results. As for the erythropoietin, which was also among the candidates, so it is appeared to be also used against situation of sepsis. So there was another drug which was on our final results, naloxone, which also considered now as an interesting candidate against coronavirus. So it seems that the findings which come up from this pure, one cell type analysis and just transcriptomics and proteomics data really resulted in a list of candidates which make sense. So, and finally, yes, this was a question about chloroquine, which is a drug. Now a lot of attention is put on that drug because it seems it's important and was mentioned. And also clinical trials are now initiated with this drug. As in our list, it was not at the top, but it was number, I think, yeah, number 8, I think. So here, after the Anakinra and other drugs. So, and again, I put here this warning that it should be taken with care. It's just pure predictions. But interesting that this drug, one of the drug target of the chloroquine is actually TNF alpha, and that's why it appears also in our list. Good. And then just to finish with this slide. So what was done is with the help of this tool called Genome Enhancer, so which you can get access through when you go to our website, of geneXplain company, together with other databases which we provide, like TransFact, TransFast, and Human PSD, and also the chemoinformatics tools which I mentioned. This one was mentioned, PAS. So with that, thank you for your attention, and I'm now open for your questions.